mutants! Many mutants! Don't worry, they're just protein constructs. So I've talked a lot about how I've done like 750 something whatever SCS pages, but I've also made a ton of protein constructs, or so these are like different versions of proteins that I want to make. And so in each of these little tubes is like a plasmid, so it's like a circular piece of DNA that has inserted into that DNA is the instructions for making a protein that I want to make. And then I can make changes to those instructions using a technique called site-directed mutagenesis. And I'll talk about how we do that using like SLIC or QuikChange. But the idea is that you make a change to the protein. So maybe you do a substitution, so you change a protein letter, or you do a deletion, so you remove a section of a protein, or you add on a tag, so you're like adding something. You do all that to the DNA recipe for the protein, and then you get the cells in recombinant protein expression, so you stick that plasmid into like bacteria and say, okay, make this protein for me, and then I can test and see what effects those mutations had. And it's a really powerful way to learn about what parts of a protein do what, and to test ideas about what you think might be going on based on like structures, so like crystal structures from like crystallography that I've talked about. So site-directed mutagenesis is a really powerful tool. And so like I have like I have 175 constructs now. And yeah, I have them in the whole spreadsheet and how I made them and all sorts of stuff. But like the reason why you have so many constructs is you can have constructs for different proteins and then different versions of those proteins. So construct is kind of this like confusion term because we can kind of use it to refer to like the DNA version that we make or the protein that's made from that DNA version. But basically it's like some sort of like edited version or some sort of version of it. So like if we could have like a truncation construct where we removed a part, or like a part with the substitution. And so we have this different like naming, like nomenclature that I'll go into about how we like represent like deletions with like delta signs, and how we like do like something like Q blah 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 A if we're like changing a Q to an A. So an A is an alanine and it's kind of like blah, like it's kind of like the most generic amino acid, so a protein letter. So sometimes people do like, you often do like a change to an alanine because it's kind of like not gonna have like, it's kind of just like the most generic thing to stick in there to see if that residue is important, but that might not always be the best change, and we'll talk a little more about that if I don't forget. But yeah, so basically using these techniques I'm going to talk about, so I'll talk mainly about like SLIC and QuikChange. There are other methods as well, but you can make these changes to the DNA and then you have a new protein construct. And so then for each one, basically it's a way to kind of like refer to your different things, and then we like, so it has a construct, and then like the original one is the wild type, and then each of the different ones is construct. And so construct is another one of those terms that kind of like confused me when I was getting into things. I'm like, what the heck is a construct? And now I'm like, I have 175 constructs. So yeah, so let's go dive into what some of this is. So I'll talk a little bit about like the background. So what exactly, like starting with recombinant expression, just a really brief thing, and then like different types of mutations, how we name these, and then how we make them. Okay, so let's go. Well, I like to think of it a bit like a Swiss Army knife. So proteins, we can get an idea about what proteins like look like using techniques like structural biology techniques like X-ray crystallography, cryo-EM, NMR, that sort of thing. And that gives us ideas about what different parts of a protein might do. And even if we don't have structural information like on that specific protein, we can do things like look for conserved regions like in the sequence or whatever that we know have some function in a different protein. And basically in all these cases we want to test if that like theory is right. So like, does that thing really do what it looks like it should do, or what we think it might do? And so we want to make changes to the protein to see if it really does do the thing that we think, and maybe we'll find something unexpected. And so yeah, so you can think about like different regions of proteins being kind of like, so different regions of proteins we often call like domains. So sometimes we talk about like certain like structural domains, which like is a part of the protein that'll fold on its own, or there can also be like functional domains. And so sometimes when the constructs that we have are like only certain domains of a protein, or like take away a domain and see if it still does that sort of thing. And sometimes the changes that we make are smaller changes, like just to change like a single letter, single protein letter, to see if that letter is really crucial. So we'll talk more about these things, but those are some of the examples of the type of things that, like, why you might want to do this. You can also do it to, like, for, like, extracrystallography, to try to get a protein construct that's actually going to crystallize. So sometimes if there are really floppy regions in a protein, you can take out that region and it'll help it crystallize. So crystallization, it requires the proteins to all, like, freeze in the exact same, like, shape or, like, orientation in it, or like this like repeating unit or whatever. And so if the protein's really floppy and stuff, it's not going to want to do that, but if you take away that floppy region, it might. So there's a bunch of reasons why you might want to do site-directed mutagenesis, and so let's talk more about it. Okay, so first let's talk a little bit about the terminology. So in order to, let's start by like going back to the beginning. So basically, so we're all on the same page with these terms. So your gene is the DNA version. Then to make a protein from it, a messenger RNA version gets made. So this is an RNA version of the gene that has like the introns removed and stuff. So when we're doing recombinant protein expression, we don't really worry about these introns, because what we're putting in is actually like a DNA version of this messenger RNA. So we use like a cDNA, so it's like complementary. Yeah, so it's like a DNA version of the processed mRNA. So we don't have to worry about the introns. But basically, you put in the DNA, and then it gets made into RNA, and then the ribosome goes along and it makes protein following the instructions in that. And so if we make changes to that DNA that we put in, then we can make changes to the protein that we get out. So when we're doing site-directed mutagenesis, even though what we care about is the protein, usually, at least in my case, like what we're actually mutating is the DNA. And so this may or may not lead to changes in the protein, as I'll talk about later, because some of these like 3-letter words, like so those spelling 3 DNA or 3 RNA version letters like spell a single protein letter, so an amino acid, and so sometimes there's like redundancy, so the same combination can spell the same thing, but we won't worry about that now. But basically, so in recombinant expression, we then put that version of the DNA version of the mRNA into the cells, and then the cells make mRNA from it, and then make protein from it. And so we call it recombinant because we're recombining like the genetic instructions for the protein into a piece of DNA, like a vector that'll serve as a vehicle for putting it into cells. And so then we have this plasmid, which has our information about what protein we want them to make, and then it has the instructions for like the cells to actually make the protein and to copy this plasmid and all that sort of thing in the rest of this like generic backbone part. So that's what we call it, recombinant. But basically, so going back to the idea of what we're actually changing. So we're changing the DNA, and then when I talk about amino acids, so those are the protein letters. So basically, when you have that string of mRNA, so the mRNA copy that gets made from the DNA, the ribosome goes along and it uses those instructions to piece together these amino acids, so these protein letters, into these long polypeptide chains, which fold up to give you a protein. So when we're talking about like a substitution, what we're talking about is changing one of these amino acids to a different amino acid by changing the gene that we put in there, so that a different amino acid gets added. And so that's like a point mutation. We're changing a single thing. And so we can do this. So amino acids have like a bunch of different properties. So they have this generic backbone, and then they have these unique side chains, so these R groups. And so some of them are really small and flexible. Some of them are big and bulky. Some of them are negative. Some of them are positive. Some of them are hydrophilic, so they're water loved. Some of them are hydrophobic, so water excludes them, and they tend to be in like the center of a protein. So when you're making a change, you have to think about like what type of change you want to make. So I talked before about how alanine was kind of like the generic one. So alanine has a CH3 group here. I don't think I have that in this slide, but alanine has just like a CH3 group here. You might be wondering why you don't do glycine. So glycine is like the smallest, simplest amino acid. Its side chain is just an H group. And because it's so small, it's super flexible, and so it can actually take on a lot of different angles that the rest of the amino acids can't. So there's 20 different like amino acids, like genetically encoded normal ones, and yeah, so glycine, it can take on these weird, like, it's more contortion-y, so it's not a good like generic example. And so you also want to use like tryptophan as your generic example, or but sometimes you do want to see like, is the size important there? Is the size of this residue important? So by residue, what I mean is that when these amino acids join together, they do so using their amino and acid parts. And so then once they're joined together, they're no longer amino acids. So we call these, what was an individual amino acid, we now call this a residue. So this is like our first residue or our second residue. So maybe this is a glycine and this is a tryptophan. When we're talking about when we want to make a change, so say we were wondering if the charge is important, to see that, so we wouldn't want to change like this this aspartate to alanine, because like, well, we might, but if we wanted to see whether the charge was important, the actually best thing would be to change it to an asparagine, which is like the same length or whatever. So I mean, it's a slightly different size and stuff, but it has this nitrogen here. It's not charged, but it's closer in appearance and properties to the aspartate than the alanine would be. And so maybe we also want to see if like the charge is important, we can change it to something positive. So these are the sort of point mutations that we can make to test different ideas. So those are substitutions. And so abbreviation wise, we do it like how we represent this in our like construct naming. We usually do it so we put the original letter first and what's changed to afterwards. And so this is often using like the one letter abbreviations. So a glycine, and we saw the 3-letter abbreviation was GLY and the 1 letter is G, and then alanine would be ALA and A. So if we changed a glycine to an alanine, we would write that as G91A. So we're saying the 91st amino acid in that chain, or that residue, I guess at that point, we're changing it from a G to an A. And to do this, we only have to change a single, we change like somewhere between 1 and 3 letters, depending on what the sequence originally was, because remember, 3 DNA or 3 RNA letters like spells a single amino acid. So we'll talk more about the types of changes that we might have to make that, but that's like the smallest sort of change you can make. You can also do insertions where you're like adding some letters. So if you can do this like in the middle of a protein, or you can like add on like a tag at the end, various things. Deletions where you're removing things. With deletions, we often indicate it with a delta sign. So we use this delta sign, so it's like a Greek delta, so that's like to show where the change was. So it's like a capital one, so it's like a triangle thing. So basically, we would write like delta 80 to 100 if we removed 80 to 100. And so maybe we have like protein X and then in parentheses delta 80 through 100 or something. So we can also talk about truncation. So we typically use if we have like a truncation like at one end, so like if it's like at the starting end, we call it an N-terminal truncation because that's like the N-terminus, or you can have a C-terminal truncation where it's like the C-terminus. And so sometimes what we're doing with these deletions is we're like removing domains like we talked about before, to see what those domains do, or maybe to a lot of times for like crystallography and stuff, sometimes it's easier to get a single domain or a couple of domains to crystallize than it would be to do the full length. And so with crystallography, you often make a ton of constructs because you're testing different truncation points, you're testing different changes, and that sort of thing. And then as we talked about, so we refer to the original version as wild type, sometimes abbreviated WT, and the change version as mutants. So yes, we really do call that mutants. That's not just like a bumbly biochemist thing. And then we can also call these different forms constructs. And so, especially like when we're talking about things that it's like a truncation or whatever, so you're not really mutating the actual stuff inside of it, but we're chopping off part of it. And so, or maybe we have a different tag on the end or that sort of thing. So this would all be different constructs, even if it has the wild type protein within it. So those are a couple terms that you might see. So going back to the changes. So when we go back to the idea of translation, so basically how it works is that you have this mRNA sequence, and then this ribosome's going through, and it goes codon by codon. So a codon is like a three-letter like block or chunk or whatever in the RNA instructions, and then this transfer RNA molecule that has an anticodon that complements that codon. So if it's like it has a G for a C and that sort of thing, and then it's going to come and it's going to bring the corresponding amino acid, and then it'll get added to the chain in this ribosome, which is a complex, and then the complex moves to the next codon, so it goes codon by codon. So when we talk about this genetic code, it's not overlapping. So you have three letter, like it's not overlapping. It knows where to start because it's signaled by the start codon AUG. But basically, so the code is degenerate, which basically it means that multiple codons can spell the same amino acid, but it's not ambiguous. So only one codon will only ever spell a single amino acid or stop. So you can look at a codon table to see like the different combinations, the different three-letter combinations. So you can see the degeneracy where you have all of these spelling serine, but it is you don't have, this will always spell serine. Okay, so now we talk about mutations. So in terms of like natural mutations, mutations can happen naturally, and so actually a lot of the early studies on proteins and that sort of thing, genes and the protein, like study all of, like original genetic studies and that sort of thing, they would make like random mutations, so like just zap flies with like x-rays or chemicals and that sort of thing, and see like get random mutations to form. But the thing with site-directed mutagenesis is we can make those mutations specifically. We could also like find natural mutations that occur and recreate those mutations. So say you have like a patient sample and they have this mutation in the gene for a protein, and then we can make that, use site-directed mutagenesis to make a version of that protein that has that same mutation, and then test if its activity is altered. So not all mutations cause amino acid changes. So you can have a silent mutation, where you have like a codon letter, one of the letters or multiple letters in the codon changes, but the amino acid doesn't change because you have multiple codons that spell for that same amino acid. So we call that a silent mutation. And there are a couple of versions when you might want to use a silent mutation. For example, like with codon, for codon optimization, when you're trying to express something, so some organisms you prefer a different codon, so they can use any of them, but they keep like more of the tRNAs that correspond to a certain one of them. And so if you change the DNA so it has that optimal codon for that organism, it might make it better. But typically you're not going to use like site-directed mutagenesis to do this for each of the different sites, because that'd be really inefficient. But you can, like, companies will synthesize it for you if you choose to go that route. But one time that you might want to do it like at a single site is if you're doing something like analytical restriction enzyme digest, or if you're doing some sort of cloning or whatever, where you need to have a cut site, a specific cut site. So restriction enzymes are these protein that recognize specific sequences in DNA and cut them. And so with restriction digest, it's a way to see if you successfully put in that like protein instructions, so that insert that you wanted to in there when you're doing that cloning. And so if you have a cut site in the insert, then if the insert isn't there, you won't get a cut there, and if it is there, you will get a cut there. And so that can help you then see if the insert is actually in there. But sometimes your like insert, so the protein instructions you're putting in there don't actually have the cut site that you want. But sometimes you can do a silent mutation to change the spelling so that it still makes the same protein, but you're changing the spelling so to a spelling that the restriction enzyme will recognize, because it's recognizing the DNA and not the protein level. And so that's a time where you might want to make a silent mutation. Then like a missense mutation, so we call it a missense mutation if it like happens naturally or whatever. But when we do it ourselves, we call it like a substitution typically, because we're changing we're substituting the protein letter. We're like doing it specifically and that sort of thing. So then you can have indels, so insertions or deletions. So this is the type of typical nomenclature and stuff that we typically use when those occur naturally. And then we talked about how we talk about like substitutions, insertions, deletions, and the type of nomenclature that we use to describe it when we're doing the cloning. And so how do we do this? How do we make these changes? So we talked about why these changes are important. So the way that I typically do it is with the SLIC method. So SLIC stands for sequence and ligation-independent cloning. And so basically the ligation-independent part. So ligation is when like you use like a DNA ligase to like stitch together like broken ends of DNA or whatever. So basically this is ligation independent, meaning that we don't do that part in vitro. We don't do that part in the test tube. Like we have the bacteria do the stitching for us. So some methods that use ligation, like so some mutagenesis methods or cloning methods and stuff, you actually need to do the ligation step. But with SLIC, you just put it in bacteria and they'll do it. And it's also sequence independent because you make these specific primers you can make to any sequence in there. So you're not, you can do it for anywhere in the plasmid. So anywhere in this construct or whatever. So basically, the idea is you do it with PCR. So we've talked about PCR in past videos, but it's polymerase chain reaction. And basically use these small pieces of DNA called primers to specify like the start and the stop of region of DNA that you want amplified. So you want it to make copies of. And so with SLIC, there are different things to do with depending on what you're doing. But when you're making if you're just doing like this change. So what you want to do is you want to make a primer that has it covers the region, so it has that specific mutation you want, but then it has parts on either side that match the DNA construct. And so this way it makes a copy that has the mutation, but it also has part of this part on like the other side of the mutation. And this is really important. So typically you do like about 20 base pairs on either side. And when you do this, so you make copies, and so you need to make copies of both strands, so you have a primer for each strand. And so you're making copies of both parts. So you have one that includes the mutation and goes in this direction, and the one that goes in the other direction. Okay, so now you have these pieces and they have the mutation, but they have and they have extra on either side. Then what you do is you add DPN1. So it's this, we'll talk more about this part later, but basically it chews up the unmutated template. So this part, so if it's not changed, it'll get chewed up, so you don't accidentally transform it and get the bacteria to make it and then think you were successful and you weren't. Then what you're going to do is add T4 polymerase. So you might be wondering why we're going to add a polymerase. So polymerase is like a thing that adds DNA letters, but it turns out when you don't give this polymerase letters, because it has this like proofreading function, it'll like start chewing away the ends. So it's going to chew away the ends to make single-stranded overhangs. So we give it like half an hour to do this. And then what we're going to add is we're going to add a single DNA, so a single DNA letter. So like maybe we only give it T or we only give it A. And so it'll then stop chewing and it'll go and start putting in that letter, but it'll get stuck if there's a different letter, because it can only put in that single letter. Like like it could put in like 2 A's or whatever if you give it A's, but then it has to stop because it doesn't have any T's or G's or whatever's next. So in this way, it chews away just enough to give you these like single-stranded overhangs, and because you have that 20 overhang or whatever, it's not going to chew away the mutation. And then you can stick this into bacteria, and they're going to actually like stitch it together for you. And so it looks like you do this, how you do this, you're getting the mutation twice, but what it's not really doing is like, it's not doing simple ligation. What it's actually doing is homologous recombination. So it's going to kind of see the regions that are the same and merge them appropriately. So it's not doing ligation. It's doing this homologous recombination. So that's why it looks like you have 2 of these things, but really it's going to, the bacteria are going to connect it into this one thing. Okay, and so that's SLIC. A different method is QuikChange. So here you're making, instead of 2 things, you're making 1 thing. So you're going to one double-stranded thing. So it's like 1 long piece. And so you use a single primer for each strand, each of which has the mutations. And then the DNA polymerase is going to copy all the way around. But when it gets around, it like stops, like it can't go through circle ever. So you're going to have this nicked DNA because it's starting at this like end of the primer and it's going around. And then it gets to the part where the primer end was. And so then you're going to have an end there. And then the bacteria will seal that end when you put them in. The way they seal it is actually with this thing like nick repair. So because the cuts are staggered a little, the bacteria will be able to repair it like it's just nicked. So it'll like still like stick together. And so you don't have to do like the actual ligation. The bacteria will use this nick repair process, which is important because then you don't have to add phosphates to your primers. So I'll talk a little more about that at the end, but something that you have to do with the like ligation methods. But so unlike with SLIC, this isn't actually like PCR because there's no amplification. So you're only going to get 1 mutant copy per template because you have that linear product. And so you can't like do the same amplification as you would in the other version because you're like starting with this primer ends or whatever. But you can do it. So it's not like as efficient or whatever. With either of these methods, you're going to want to do this DPN1 digest. And so basically what happens so you have this plasmid that you put in, so it has the thing that you think you put in, and then it has like a selection marker, so it's like an antibacterial resistance gene. And so you use transformation where you like stick the vector, stick the plasmid into the bacteria and say, okay, make this for me. Well, typically at that point you just want them to make, instead of making the protein, you first want them to just make lots of copies of this vector, this plasmid that you put in there. And then you can check it to see if it was actually the right sequence using like analytical digest, and or then doing like sending it for sequencing to make sure the spelling's all really right and stuff. But when you're doing this mutagenesis, because you're starting from this plasmid that's originally there before you changed it, and so both the changed and the non-changed version are going to have this resistance gene. So if you just stick it into bacteria, the parent vector, so like the unchanged version, is still going to have the ability to grow. And it's going to have a growth advantage because it doesn't have to be all healed and stuff. And so you want to make sure that you remove any of that parent plasmid. And the way that you can do this is because those methods use like PCR or like chain extension or whatever, those methods that are doing it like in the test tube, they're not going to have this like methylation. So when the DNA is copied in bacteria, it gets this methylation. So that add these like methyl, like these CH3 groups to it as part of this process. And so this restriction enzyme DPN1, it actually recognizes like methylated DNA, and it's going to like chew that away. So it gets rid of the parent, but it won't get rid of the changed version. So you want to do that before you put it in. Okay, and so yeah, so that should help with that. Another thing to keep in mind is that if you're using a like ligation-independent method, where you're actually like trying to do some like stitching in your test tube and that sort of thing, if you're doing it with like a primer based method where you have like a primer that you're extending and then you're stitching it together, the primers, when you buy them, they typically don't have a phosphate group at the end, which can be a problem if you're trying to ligate it, because you need that phosphate group for that ligation. And so you need to add that on beforehand with like a phosphate, with the kinase, that you can do like a T4 polymerase kinase, or that sort of thing. Or you can buy it phosphorylated, but then you have to pay more. But anyway, so hope that helps. And you can find a text version on my blog, and I will stick in a link, and it has a bunch more details about these various methods and various concepts. And so, yeah. And now it's time to get back to mutating my thesis, hopefully constructively.
Video này do mình, Cương bên npslab.vn, từ chỉnh sửa phụ đề hoặc lồng tiếng bằng công cụ, thấy vậy thôi, chứ tốn công lắm á. Thấy hay thì nhớ bấm theo dõi kênh, cho mình thêm động lực làm mấy video phòng lab xịn sò nữa nha.